Well, I want to start with two comments. First, I'm extremely grateful to have been invited to this symposium, which I've enjoyed enormously, and to be back in Seattle, which is always a treat. And second, I have to make a confession that I'm one of those exuberant, out there, small idea scientists who's going to focus down on a single process, and then at the end come back to systems neurobiology. So I want to tell you about a really remarkable process in the brain, which is responsible for setting forth the laminar structure of cortical regions of the brain, the neocortex, and my favorite region of the brain, which is the cerebellum. So I'm going to tell you a bit about a fundamental process that's responsible for cell systems, that is, how you build these systems in the brain. Now, I think it's a great tragedy that in recent years the beauty of the cerebellum has been lost a bit at the expense of the frontal cortex, because the cerebellum is a truly magnificent structure. It's the simplest of the cortical regions. It predates in evolution the neocortex, and it has two fundamental cells, the granule cell, which is my favorite. There are 45 billion granule cells in the human brain. That's more neurons than there are in the cortex, and as you'll see, this is important for the studies we're going to do. And this cell connects to the Purkinje cell, which is the single output neuron of the cerebellar cortex. Moreover, it has an unusual developmental history, which I won't go into, but this granule cell gets born in an ectopic layer along the surface, which means that its cell neurogenesis continues well into the second postnatal week in humans until they're 2 years old. And this gives us a really beautiful model system for studying glial-guided migration, which is the essential step in making cortical lamina. The exact same process happens in the cortex. Here are a few slides of the different systems you can use. Here's Pascal Rakic's classic image of the neuron migrating along the glial fiber, showing the intimate relationship. The nucleus is in the back of the cell, and so on. So this is the cortex from one of our papers. And the beauty of the cortex is, of course, everybody's interested in the cortex. The difficulty with the cortex is how complicated it is. There are dozens, if not hundreds, of types of cells in this system. The cerebellum allows you to study the cerebellar granule cell. And so what we've done over many years, and what I'm going to subject you to for at least 15 of the 25 minutes, is a detailed analysis of this migration in the granule cell, which one can, of course, then use to go back and look at cortex. So the genetics, one would like to identify the genes involved in migration, which, of course, we've done. I'll talk about that in more detail. The staggering number of granule cell progenitors. You can get 10 million from a single litter of mice. You can make cDNA libraries. You can make defined cell recombinations and so on. This extended period of neurogenesis, and last but not least, the granule cell is thought to be the cell of origin of the most prominent pediatric metastatic brain tumor, the medulloblastoma. So that's my pitch for the cerebellum. Over many years, our lab has studied glial-guided migration. I'll give you a sort of bird's eye view of the fact that glial-guided migration is a specialized form of motility. It does not work by the leading edge-based type of motility that the growth cone, which is a fibroblast on a string, works. And I'll show you over the next few minutes that there are perinuclear motors and control systems that control this essential process for brain development. Over a number of years, we've used purified cells from cerebellum and recombined them with glial fibers. At this magnification, the glial fiber would run down to the lake. And we also did a lot of cell biology experiments some 20 years ago, where we purified neurons from one brain region and took them on a walk on glia from another brain region. And with that type of approach, we were able to prove that it's more like that this system works like the interstate highway system, with the neurons like the cars having the navigational instructions in the neuron, and the glial fiber system, as important as it is, being a passive system that the cells migrate along. This is an old movie, but I'm showing it just for fun because when we did it, there was no web. And now, since we can take the whole 6 gigabyte movie off, we can see the real details of migration. As the neuron moves, instead of a leading process, there are tiny little lamellipodia, just long enough to wrap around the glial fiber, and the neuron forms a junction underneath the cell soma. So here's the neuron here. If you look closely, you can actually see the centrosome that I'm going to talk about again in a minute. The adhesion is underneath the cell soma, and what's going to happen is the cell is going to bind, then let go, and glide along this every 3 minutes. So this we studied in the 80s. Then over the last few years, with systems-type approaches of microarrays, we found this gene PAR6, which many of you will know as a conserved polarity protein, that was upregulated when the cells started to migrate. And when we expressed PAR6, and this is under, you can purify the cells, and because they're dividing, you can use a retrovirus to put anything you want in them. So we put PAR6 using Venus, not EGFP, 20 times as bright. You can put in 1 copy of the gene and get lots of protein without interfering with function. And we saw this very adorable little motion. It's a kind of 2-step do-si-do. You'll see the centrosome labeled with PAR6 move forward, then the cell moves forward, 1, 2, 1, 2. Lots of times the cells stop. This is another subject to itself. But the main thing that happens when the cells stop is the centrosome stops moving. So David Selecki in the lab did this Herculean task of looking at 80 cells that he imaged, and he'd watch the centrosome move forward and then the cell body move forward. And once again, within seconds, accuracy to what we'd seen with adhesion is a 3-minute difference between movement of the centrosome, movement of the soma. When you look at the same thing in cells that have stopped, it's random. So it's a very controlled type of motility. It's very different from a fibroblast. And in experiments that I won't have time to show you, the nucleus is enclosed in a kind of bingo cage of tubulin. We showed that the nucleus also has dynein on its surface, so the dynein motors run down the tubulin cage. The centrosome right next to the nucleus moves forward, elongating the cell, and then the nucleus steps up behind it. So the next question we wanted to ask was to look at cytoskeletal elements, to ask, well, what's the force behind this? Because those of you who follow systems biology will know that many of the genes that have been identified to be involved in cell migration are indeed components of the cell cellular cytoskeleton, this one perhaps being the most famous. So I'm going to show you I've showed you about the perinuclear centrosomal movement, and so now this is PAR6. This is completely out of place, I beg your pardon. The beauty of this molecule is that it's a signaling protein that controls the activity of an atypical kinase. Its upstream regulators are the Rho GTPases, which are, of course, the polarity proteins of vertebrates. And so the other thing that's exciting about it is that where there is 110 bricks and mortar in the centrosome, this is the only bona fide signaling pathway protein in the nucleus. So we wanted to use genetic techniques to knock it out just in the centrosome, but we couldn't do that because when we did a FRAP analysis, which is to fluorescently ablate something and then watch what the turnover rate is, the turnover rate was 30 seconds. So it's not only a signaling protein, but it's turning over very rapidly in the nucleus. It's also involved with microtubule dynamics, as you can see by labeling the tips of microtubules with EB1. In the normal cell, this is streaming out like the 4th of July. If you put in too much or too little PAR6, you get this very sad little picture over here, where only a few microtubules are coming out of the centrosome. So we wanted to look at what happens if you perturb the upstream regulator, CDC42, the Rho GTPases, particularly CDC42. And I'll just tell you this briefly. If you activate CDC42, the neuron runs faster along the glial guide. If you use a dominant negative or an shRNA or genetic techniques, you get this bad hair day looking cell where it's got multiple processes, and as you might imagine, this does not migrate very efficiently. So the PAR6 complex and its upstream regulator, the Rho GTPases are very important. And I'll just quickly show you the cytoskeletal dynamics are important, and it's no big surprise, perhaps, that the actomyosin motors are what control the actual force generation in migration. There are lots of really beautiful ways to image these cells. These are tiny little cells, 4 microns in diameter. You can make a histone fluorescent, or you can make the actin fluorescent or the centrin centrosome, other markers for the centrosome, and look at a lot of details. And you can also use this centrosome as a kind of indicator of what the cell is up to. So here the centrosome is moving along, the cell is moving, and here we use an inhibitor of F-actin. It's actually a stabilizer, so it perturbs the dynamics of F-actin. And what you see is as soon as you add that, the cell stops migrating. You can wash it out and turn it around. This is a surprising picture because we always thought the actin would be behind the cell, pushing it like a tube of toothpaste. And although there is actin in other parts of the cell, you see this glob of actin in the front of the cell, down near where the centrosome is. Oops. We also looked at myosin motor, fibril, and we could use an inhibitor of that and show that this also perturbed migration, or you could do it the other way around and take calciculin, which jumpstarts myosin fibril assembly, and show that then you could send the neuron migrating down its way. So finally, in this series of things, if you perturb PAR6 levels, you also perturb assembly of the myosin fibrils by perturbing phosphorylation of the regulatory light chain of myosin. So we could understand all of this in a lot of detail, and we're very excited about it because over many, many years we've sought to explain exactly what's special about glial-guided migration, exactly how it works as compared with other forms of motility. And basically, as I told you, you've got the centrosome moving forward, the nucleus being restrained and in this little tubulin cage. And then you've got these actomyosin motors, which I'm not showing you this, but they connect to the adhesion site, pull against the adhesion site so that it will be released as the cell moves along. The one fun thing about having postdocs, there are many fun things in your lab, is they get to name the models. There are actually these guys, there are many insane things in the United States, but one of them are these guys who go around pulling tractor-trailer trucks, trains, whatever, and they call themselves strong men. So we adapted this to show the strongman model, just to show that even though it doesn't seem to make a lot of sense, that pulling from the front can get a cell down the fiber, and is actually how glial-guided migration, this essential form of migration that's so important to developmental disorders, to epilepsy, and so on, works. So many, many years ago, we looked at the adhesion proteins that were involved in migration, and we described one, gave it a bad name, astrotactin, meaning neuronal protein for touching the glia. And we published many papers on this and showed that this was essential for migration. Since then, all of the other neuron glial adhesion proteins, mostly integrins, have been knocked out, and none of them actually function in migration. So astrotactin is the last bona fide standing neuron glial adhesion protein. I'll skip this as some fancy imaging of that. And the exciting thing was we found that there's a second astrotactin that looks all the world like the first. I'm getting to systems biology, don't worry. The second one co-localizes, this is with row B in endosomes, as the cell is migrating along. The beauty of the cerebellum is also that these cells are born out here in this zone, and then you can do a simple assay and watch this migration process, which is so much more complex to watch in the cortex. And when we did this, we could show that a small molecule inhibitor of endocytosis called dinosaur, we did not name it that, inhibits the movement of the cells from the outside to the inside, as you can see here, and it's reversible within 10 minutes. So what's going on? Oh, there's astrotactin 2. Astrotactin 2 is a huge gene. Unlike astrotactin 1, which is only expressed during the migratory period, it's expressed in the adult brain. Moreover, it's in cortex and hippocampus, and I'll tell you more about that. Here's a comparison of the two. They look for all the world the same. We're doing structural biology with Gunnar van Henje's wonderful group. It's a very strange protein. The N-terminus is outside. There's a completely useless cytoplasmic domain that doesn't interact with anything as far as we can tell. And then there's the C-terminus comes back outside again. This just shows that unlike astrotactin 1, which is a bona fide adhesion molecule, astrotactin 2 in the membrane, but not in the cell surface. So the last little bit I'll show you about this is if you use flow cytometry, you can say, well, what happens as you change the stoichiometry of astatactin 1 to 2? Is there any point of 2 versus 1? Here's neuroligin, where you see lots of the protein on the cell surface. Astatactin is not as much of it is on the surface as neuroligin, but what happens as you titrate in astatactin 2 is astatactin 1 disappears from the surface. So astatactin 2 is not a neuron glial adhesion protein, it's regulating the trafficking of the first one. So here we have a situation where the adhesion molecule is, astatactin is chewing its foot off basically by regulated endocytosis, controlled by the PAR6 protein and the recycling dynamics of astatactin 1, which are controlled at least in part by astatactin 2. So we don't imagine that astatactin 2 was put on the surface of the earth just to regulate astatactin 1. So we did the classic experiment of an IP and mass spec. We're finally getting towards systems biology, and we found all these interesting proteins. I'll point out one of them. There are two of them here that are interesting. Complement C1q, the subcomponent A, Ben Barris and others have published, is involved in synaptic plasticity. Collapse and response, the same. The rest, which I'll show you in a drawing, all seem to be involved in the nuts and bolts of receptor trafficking, clathrin-mediated trafficking, which of course the synapse uses as well. So astatactin 2 is binding to these specific components of these myosins, for instance, are only involved in receptor trafficking, vesicle trafficking, also directly to clathrin and transferrin. So astatactin 2 is either involved in trafficking per se or is carried along by the system and therefore picks up all of its components. Interestingly, we've looked at other adhesion proteins that astatactin 2 interacts with. The most interesting, I don't have time to tell you about it, is incadherin, but it doesn't interact with all adhesion proteins, a subset of them. So let me just go into what's going to be of more interest probably to the systems people. We've looked for many years at the development of the granule cell and described genes that were important for this. And the one thing I forgot to tell you is, unlike astatactin 1, for which there are no known human mutations, there's more than 10 papers in the last year showing that astatactin 2 is associated with the great developmental diseases, autism, ADHD, and schizophrenia. So what we've wanted to do is to begin to be able to study human cells with iPSCs. And being a little on the OCD side, we would like to be able to design experiments where we could make the particular neuron we want and not some generic cortical neuron. So as an approach to this, we've taken, whoops, missed the slide. And here the we is my colleague Nat Heinz, who's pioneered bacterial artificial chromosome methodology. And I'll show you this slide and then come back. He and I were co-PIs for this GenSat project where we made 600 lines of transgenic mice that express EGFP under the control of a BAC. So by homologous recombination, you can pull the BAC vector in just before the start site. It's, for us, at least, we think, preferable in many ways to typical promoter-driven transgenics. The BACs are bigger, so you have regulatory elements at both the 5' and the 3', and to get stable expression of the gene. So there are 3 levels of this. EGFP is the one we did for GenSat. We also made 100 lines of Cree mice with this approach with a Cree in here. And then recently, Nat and Paul Greengard had developed this BAC trap, or EGFP, fused to the ribosomal protein L10A. So you can get a nice marker for either our favorite, the granule cell, or the Purkinje cell, PCP2. Here's the granule cell marker. It's NeuroD1 gene. So now in this mouse, what you've got is the EGFP fused to a ribosomal protein. So you've got green ribosomes. You can use an affinity matrix. You can just use antibodies to pull down the ribosomes, pop off the RNAs. then you can do RNA-seq or an array or whatever you'd like to look at this. So this is very exciting because it allows you to interrogate gene expression at all ages from development when we can purify cells on. And here we've done the analysis of these experiments with Jill Miseroff at the Broad. And I won't go into them in any detail, but it is very exciting that as you go through 5 different ages from P0 to P56, the heat maps show different types of proteins of functions being expressed. We know a lot about this gene set, and we want to use this gene set in order to directly compare native cells with differentiated ES cells, because we believe this will provide a more rational method for inducing the cells to be the ones you want. So what we did was we took the backtrap mice that Nat generated, the transgenic mice. Then we generated ES cell lines from the backtrap lines, differentiated them into cerebellar neurons. I won't bore you with the developmental biology of the cerebellum. We used a strategy, which is basically proof of principle of the development, to first induce the cerebellar territory along the AP axis, here engrailed 1 and 2. Kathy Millen's in the audience who discovered engrailed 2. And then to induce granule cell specification. The BMPs are very critical for this. And then pathways that differentiate them into a fully mature granule cell. What's going on? So the way we do this is to follow much of what's known from the development that we studied for many years. I won't bore you with that, but it's a coherent approach. And the nice thing about it is we were able, some years ago with the first mouse ES cells that we did, to set up an implantation into the cerebellum to see if we could get the cells that were differentiated from these transgenic mice. And here they're green, migrating into place perfectly. And they go deep to the Purkinje cells and make dendrites just like they're supposed to. But some of us are curmudgeons and wonder how close are they really? And so we took the ones from the backtrap transgenic mouse. And we then compared the 5 different ages that I showed you at a glance for the granule cells and the Purkinje cells with the cells, the ES cells that have been differentiated to be a granule cell. So how good are they? So here I'm just showing you adhesion proteins because many of these are markers for neurons. So here's at time 0. This is before we differentiate them. You see lots of blue down here for famous adhesion proteins, L1, NCAM, astatactin, of course, neuroligin, and many, many others. As you look at 2 different stages of differentiation of the ES cells, you see that the ES cells do quite well with turning on these differentiation genes. You can compare these with the granule cells and the Purkinje cells at different stages. And so by this measure, you would think that the differentiation goes quite well, and this actually matches with what we and many others have used, which is just markers of developmental genes to say that we've got a granule cell, for instance. But when we and the we is the Jill Miseroff at the Broad. I won't go through all of this, but I'll just say that there are people who know what they're doing with statistics to look at this, to do the cluster analysis of salient features rather than, I've got 2 slides, rather than doing networks. And what we found when we did this, as my son would say, duh, Mom, was the essential difference between differentiated ES cells, or the most salient difference, I should say, differentiated ES cells and native cerebellar granule cells. Here they are, a whole list of ion channel proteins. And this is kind of interesting because there are those of us who are developmental biologists, then there are people who are physiologists, and the twain don't meet very often. And we know very little about what controls the expression of these genes. So the challenge ahead of us is to do lots of experiments to be able to induce these. I'll just say at the very end that The mutations in astatactin 2 have gotten us into doing human studies. We've had some difficulty getting material for this, but happily Johns Hopkins contacted us because they have an entire family. The father has ADHD and has a copy number variation that's a deletion that's effectively a knockout of astatactin 2. He has 2 autistic children, and so we're just getting these cells. And the plan will be, of course, to take the skin or blood cells from these members of this family to generate iPSCs and to differentiate them into cerebellar neurons. And we've developed ways to express the backtrap in the human cells so that we can follow gene expression of 20,000 genes, not 10 markers, in these cells to be able to tell how faithfully they represent a granule cell or a Purkinje cell, and also so that we can look from clone to clone to see what the variation is. And then we can do in vitro functional assays like migration or synaptic assays. And the other thing that's beautiful about the cerebellum is, like I showed you for the mouse cells, we can implant them into mice. And with the new methods, Clarity, but I would point out that a month before Clarity came out, Carol Mason at Columbia published Clear, which also works for dye-eye, so it's really spectacular methodology. So I think that these things are going to revolutionize our ability to do genetic experiments in the mouse with labeled markers. So I thank you for your attention, and I also need to thank, the people in my lab who've done these experiments. Eve-Ellen Govek has done many of the migration experiments along with David Selecki, Keisha Zaki-Horn, Xiaodong Zhu, and Huranaz Behesti are working very hard on the iPSC and genomics of iPSCs. Nat Heinz, my longtime collaborator and dear friend at the Rockefeller, who's been involved in many of these experiments, Linda Van Elst with the Rho GTPases, Jill Mizaroff and Adrian Heilbut are helping us deal with all the data from systems biology. So thank you very much. I hope this wasn't